This video is going to talk about mitosis and the cell cycle. So, the cell cycle is the entire life cycle of a single cell. And that cell survives, it does many things, it metabolizes, it produces numerous proteins, it tends to have a job, and then it either dies or produces new cells. Now, quick disclaimer, most of the cell cycle is actually all of that other stuff when the cell is surviving, doing its job, metabolizing, making proteins, helping out whatever system it happens to be living in. But for the purposes of this video, I'm going to talk only about actually mitosis, which is when the cell gets to the end point of its job and decides to multiply to make new cells. There are a number of reasons cells decide to do this. They need more resources and they would like to produce more things. In the cases of multicellular creatures, they are often trying to replace other cells that are damaged in a different location and then rebuild them, or perhaps grow the organism. Either way, one cell becomes two. So here, in front of us, there are two cells. There is a standard animal cell, you can tell because it is circular, and a standard plant cell, you can tell because it is square, because plant cells have cell walls, so their structure is a little different. Not that all cells are these shapes. And inside each of these cells is a standard nucleus, in which you can see a large dark staining region, but you can't actually see any individual chromosomes, because when the cell is active in interphase, it is doing its job, and the chromosomes need to be loose and available to be transcribed. So this would be interphase. Most of the cells that you see in most images will be in interphase. Now, if you're looking for a really great animation of mitosis, I suggest going elsewhere. All you're going to get today is sort of my crap version of it, but it'll give you the basics as we walk through the different phases. Now, you may have heard of mitosis and the 4 different phases that are involved in mitosis. You may even have heard the sort of semi-acronym PMAT, P-M-A-T, that goes over these 4 phases: prophase, metaphase, anaphase, and telophase. I'm going to break each of them down so that we can see what happens in each phase. So starting with prophase, pro typically means early or at the beginning, and in this case that's exactly what it is, or even before, and it is the phase that is first, because it is when the cell decides it is going to definitely divide. And the very first thing that happens is the nucleus has to break down and let the chromosomes out. So in both of these cells, I'm going to remove the nuclei, and what I'm going to do instead is I'm going to add chromosomes. Chromosomes typically would be just one long piece, but during the process of interphase, the chromosomes duplicated. This happens in a section called the S phase for synthesis of DNA. And so I will draw them instead so that they are little X's, which is how you're used to seeing them, which is 2 pieces stuck together at the middle. I'll go ahead and put 3 little chromosomes in each of our cells. So at this point, the chromosomes have just broken out of the nucleus, and they're just kind of hanging around. But they are visible. They coil up very tightly so that we can see them in the staining. All right, that takes us to our next phase, which is the M1 for metaphase. So metaphase is in the middle. And what's going to happen is these little chromosomes are going to indeed also go to the middle. This is where they line up. As you can see, I have magically lined up my chromosomes right in the middle of each of these cells. The next thing that happens is anaphase, and that is our A, anaphase. And in anaphase, the chromosomes are going to come apart, which is a good way to remember anaphase. So again, I have magically taken them apart, and you'll notice that each one of them was an X, but then separated at the middle so that one piece went one way and one piece went the other way, so that they literally have a line of space down the middle of them, and the chromosomes have split apart. The last phase of this process is telophase. And in telophase, we are actually going to essentially reverse what we did in prophase. So the chromosomes are going to decoil or uncondense and become sort of loose, squishy stuff again, and they are going to form nuclei. Only since they split apart, there's going to be 2 nuclei, thus telophase. Let me make the magic happen. Ta-da, 2 nuclei. Now, if you have seen pictures of mitosis before, you might say, well, this seems kind of strange. Nothing else has happened with the cell. And that's because the process of mitosis technically only describes what's happening with the chromosomes. The process that is happening with the cell is called cytokinesis, for moving cell, and it happens at the same time as these other things are happening, but it is technically a separate process. I'm going to do it separately here so that you can see what I'm talking about. So for both of these, what's going to happen in cytokinesis is that the cell membrane, and in the case of the plant cell, the cell wall, is going to first expand to create a larger space for these new nuclei to be in. Boom, magical cell membrane and cell wall expansion. But also, cytokinesis allows for these to become 2 separate cells by separating and adding different pieces. In the case of the animal cell, the 2 pieces actually start to come towards each other in a pinching mechanism. And eventually, see, this one I will show, they will pinch together so tightly they actually make 2 cells and could pop apart. In the case of the plant cell, they can't really pinch because the cell wall is rigid, but what it will do is it will begin to build a small cell wall in the middle between them. And as the cell continues to divide and cytokinesis goes on, the cell wall will get all the way across and cause these to separate. This is also when the organelles, all the little things you can't see, like mitochondria and endoplasmic reticulum pieces and things like that, separate and divide across the cells, so you have 2 complete cells. So those are the 4 stages of mitosis, separating the chromosomes, plus cytokinesis, which separates the cell, which is how you get from 1 cell to 2 cells. So I wanted to get on here to show you a quick and easy way to also help you remember the stages of mitosis, going through all of them: prophase, metaphase, anaphase, and telophase. So if you can remember the PMAT to get the phases, here's how you do what happens in each one. In interphase, you start with just 1 nucleus and you can't see the chromosomes. In prophase, the cell prepares by breaking apart the nucleus and making visible chromosomes. In metaphase, they line up in the middle. In anaphase, they come apart. They kind of look like this too. In telophase, they make 2 new nuclei. And then you just have to remember that cytokinesis is needed to make the new cells. All right, so now the important part, which is being able to recognize mitosis when it is occurring under the microscope. And specifically in this particular case, we're going to examine these plant cells in the onion root tip. So this is an onion root tip, and this is where the onion root would be mostly dividing. Not down here, this is the cap or the part that protects it, but right in here are a whole bunch of cells that are dividing. So So let's zoom in on them a little bit. Now you can actually see the cells. You can see the structured cell walls giving these kind of a rectangular shape, and you can see all of the nuclei. Again, most of these cells are still actually in interphase, as in they're not dividing, but here and there you see a few that are a little bit interesting. As we move around, and as you move around in the different places you might be looking under a microscope, the key is to hunt down the ones that look different. So there's a couple different ways that they look different. So if this here is a pretty standard interphase cell, right next to it, you'll notice all these little lines. That would be a cell in prophase. They're a little bit hard to see and they're not perfect, but they're lined up pretty nicely. Another cell that might be in prophase could be here. Another one could be here where it's just beginning the process. So if you remember correctly, metaphase was lining up in the middle. And you actually can see the chromosomes pretty well in metaphase. If you look right here, you can see them lined up in the middle. I'm going to switch my arrow so it's a little bit easier to spot. Hey, there we go. I'm going to switch to an arrow so it's a little bit easier to see. Let's look at another section slide. So here again, mostly cells in interphase, but as you start to look towards the edges, you can see more cells in different phases. So here's one that's probably a squished version of metaphase. Over here is a prophase, and right here is a really good anaphase. You can see chromosomes here, chromosomes there, separated. Here's another one that is probably in anaphase, because you can see that they are separated. As we move on, we also want to see cells that are in telophase, where they're actually becoming two new nuclei. Can you see where those might be on here? They're a little bit hard to spot in some cases, but I've found one that I think is right here. So you can see the middle part is dark, it's just starting to form, but I see at least two nuclei hanging out there. Let's check out one more different image, because this one has some really good ones on it. And so we'll look for all of our phases. So here's all the cells in interphase that we would anticipate. Here's at least one cell going into prophase and another one. This one here looks like a pretty good metaphase, and so does that. This is another fantastic example of anaphase, where they're really well separated. And then telophase, which sometimes is one of the trickier ones to find, usually involves having to look for sort of two real small nuclei somewhere next to each other. So here, these could be it, or they could have just finished dividing. Way over on this far side is another example where it looks like there's two nuclei and a little bit of a cell wall, and it doesn't go all the way across. So that definitely makes me think telophase. Just one more image for you, because animal cells are a little bit harder to catch under the microscope in various stages of mitosis, but they still are pretty easy to spot once you see them. Prophase looks like prophase, metaphase looks like metaphase, anaphase looks like anaphase. But here, in telophase, I can point out how the animal cell pinches and forms this, what is called a cleavage furrow, to create the two different cells. So that's all I got. Go check out the microscope and find your own cells in various stages of mitosis.